Okay, so welcome to this next video in which we are discussing the contraction of cardiac muscle. Okay, so what we've seen now is that we have produced this calcium signal which is global. So we certainly have done that in ventricular myocytes. In atrial myocytes, we've seen that we produce a calcium signal around the periphery of the cell, which becomes a global calcium signal if you stimulate the atrial myocyte with beta-1 agonists. Okay, so we want to see now, how does this calcium actually cause the sarcomeres to contract? Okay, so remember, back to our picture here, here's our sarcomere. So what we want to start happening is we want these myosin filaments to start climbing up the actin filaments. Okay, so we want to now try and understand how that happens. And this is a process known as Crossbridge Cycling. Okay, right. So the beginning process then is that calcium needs to start activating cross-bridge cycling. And the way it does this is it causes the inhibition of a protein which is inhibiting the cross-bridge cycling. So if we draw our sarcomere out again, so let's draw our sarcomere here. So here are our Z discs here. Okay, here are our actin filaments here. Okay, and it's probably going to be a bit small. So here are our actin filaments attached to the Z discs. Then we have our M disc in the middle with our myosin filaments here. Okay, right. So let me colour this in so that it looks nicer. Okay, so in green, these are the actin filaments again. Okay, attached to their Z discs, which we drew in purple. So in purple, here are the Z discs. Okay, and then in orange, here is the M disc down the middle. Okay, and then off the M disc come the myosin filaments in blue. Okay, right. Now, there is a protein that I haven't told you about so far. A protein that is wrapped around the actin filaments when the cardiomyocyte is at rest, basically. When it's not contracting, there is a protein that wraps itself around the actin filaments, okay? And this protein is known as tropomyosin. So here is tropomyosin. And tropomyosin being round around the actin filaments stops these myosin filaments here from being able to interact with the actin filaments. And if you remember, in order to get sarcomere contraction, in order to bring these Z discs closer to one another, you need the myosin filaments to be able to interact and climb up the actin filaments. Okay, so this tropomyosin is blocking that climbing up. So basically what we need to do is move that tropomyosin. Now there is a protein bound to the tropomyosin, so let me show this. It's bound many times. There are lots of this protein bound to the tropomyosin, and we'll show this protein in turquoise here. So in turquoise, there is a protein that modulates the position of the tropomyosin on the actin filament, and this protein here is known as troponin. So this is the troponin protein. Okay, now what happens is when calcium goes up in the cytoplasm of the cell, the calcium can bind to the troponin protein. Specifically, the troponin has 3 subunits, and the subunit it binds to is the troponin C subunit. Troponin C, which is named troponin C because it's the calcium binding subunit of troponin. So it binds to troponin C, which is a subunit of troponin, so it's part of troponin, and basically that causes a conformational change in troponin. And when troponin undergoes this conformational change, it moves the tropomyosin slightly so that the position of the tropomyosin wrapped around this actin filament no longer blocks the myosin filament from being able to interact with the actin filament. So now the myosin filament can start interacting with the actin filament, and it can start the process of crossbridge cycling. So let me now describe to you the process of crossbridge cycling. So this is an interaction between the heads of the myosin filament. So here is a myosin protein within a myosin filament. So here is the myosin filament, and this is a single myosin protein sticking out from the myosin filament. Okay, and here is our actin filament here, made up of many actin monomers. Okay, so they should be coloured in green. So where's the green? Right, so this myosin head begins the crossbridge cycle with adenosine diphosphate, ADP, and an inorganic phosphate molecule attached to it. So at the beginning of the crossbridge cycle, the myosin head here has adenosine diphosphate, ADP, and it also has an inorganic phosphate molecule bound to it. Okay, now the first stage of the crossbridge cycle is the formation of the crossbridge. Now, this is basically a bond between the myosin head and the actin monomer, and one of the actin monomers, so one of the myosin binding sites on the actin monomers. Now, in order for this to happen, what has to happen is the myosin head has to drop this inorganic phosphate. So, basically, it's going to drop the inorganic phosphate and it's going to form a crossbridge with one of the monomers of the actin filament. So these are these actin monomers here in green. I'll colour them in so we know what they are. So these are actin monomers in bright green here. And then the myosin head is in blue here. Okay, right. Now, it's still got ADP bound to it. It hasn't given that up yet. So it's formed this crossbridge. Okay, so this is crossbridge formation here, this stage here. Crossbridge formation, and the bond between the myosin head and the actin monomer is known as the crossbridge. So this is crossbridge formation. Right, now what's going to happen is the myosin head is going to drop the ADP and it's going to perform what's known as the power stroke. And by the way, we are looking at a myosin filament on this side because I have oriented the heads pointing this direction. And remember, I told you the myosin heads are always pointing towards the actin filament, at least at the start. So they're pointing in this direction. So we're looking at this side. So what's going to happen is something known as the power stroke now, and this is what produces the force of muscle contraction. The myosin head is going to drop that ADP molecule, and in so doing, it's going to change conformation. It's going to contract back, pull back this way. Okay, and this is known as the power stroke, this movement that it makes, where it sort of folds back onto its fibrous tail. It's known as the power stroke. Now, even though it drops the ADP, it remains bound strongly to that myosin monomer, to that actin monomer which it had formed the crossbridge with. Okay, so here in green are these these actin proteins that make up the actin filament, and here in blue is the myosin head still bound loyally to its actin monomer. So what's going to happen when it does that? Well, let's look at this. This is the myosin head here, and it's now just pulled back in this direction. It's folded back onto its fibrous tail. It's folded back effectively towards the M line here, or the M disc, and in so doing it pulls the actin filament in this direction. So it's pulling this whole filament in this direction, and therefore it's pulling the Z line or the Z disc in this direction. Now the same thing is happening on this side, except in the opposite orientation. So the myosin heads here are oriented in this direction, and when they undergo the power stroke, they'll collapse back onto their fibrous tails too. They'll collapse back this way, and they'll pull the actin filament here in this direction. So this actin filament is going to get pulled in this direction, and that's going to bring these two Z lines like this, or Z discs like this, towards one another, contracting the sarcomere. Okay, now, how do you complete the cycle? It needs to be a cycle, so you need to return basically to the original state. So what now comes along is you bind a molecule of ATP. So the ATP is going to come along and it's going to bind to this myosin head here, and it's going to cause the crossbridge to cleave. So this next step is the cleavage of the crossbridge between the myosin head and the actin monomers. Okay, so here is the actin filament, and here is the myosin head, but they're no longer bound, and it's because this ATP molecule has come along and it's ruined their relationship, basically. It's stopped them from binding to one another. It's made the myosin head decide that it doesn't need the actin monomer anymore. Okay, so it abandons the actin filament in favour of this ATP, and that stage is known as crossbridge cleavage. So this is crossbridge cleavage, and now what happens is that you hydrolyze that ATP, cleavage, okay? You hydrolyze that ATP to adenosine diphosphate, ADP, an inorganic phosphate, and what that does is it returns the myosin head back to its original conformation, so it moves back out. It returns to its original conformation, but the actin filament is now still over here. That original portion of the actin filament that it bound to, it's now moved in this direction, basically. Okay? So here's the actin filament. So now it will be neighbouring a piece of the actin filament that's further along. So here is a further along portion of the actin filament, so some new actin monomers to interact with. Okay, and now once it's hydrolyzed that ATP to return back to its original conformation, it now has ADP, adenosine diphosphate, bound to it, and also an inorganic phosphate. So effectively it's back to the original state that it was, except that what has it done? What has it overall achieved? It's basically shifted this actin filament in this direction completely, because these original actin monomers, which were directly above it, have now been shifted to this side, and that's effectively moved the entire actin filament in this direction, and that's happening on both sides. So you are moving these two Z discs towards one another, and that's the crossbridge cycling, which occurs within which occurs to contract the sarcomeres. Okay, and we'll continue our discussion in the next video.